Good afternoon to everyone. First of all, it's my pleasure to introduce Dr. Galia Avidan. Galia, special pleasure to introduce her because first and foremost, Galia was my first PhD student, so personally it's really a great pleasure. And not only that, she is from one of the first cohorts of the ICNC. This is the third one?
I think it's the third.
The third one. And
you start calculating.
I overheard one of the female students here talk about, the women getting position in universities. So first of all, Galia is not only at Ben-Gurion University, but she's already tenured. And so if you are looking for a role model, here you are. And it's really an honor and a pleasure to have you here, Galia. And we're looking forward to hearing your talk.
Thanks, Rudy. And thanks for inviting me. And now I feel really stressed because I have to be a role model. It's a big job. So I'll try to talk about face perception, and let's see if I can do that. So the talk I'm going to give today is about research that I've been doing for the last several years. It's work that some of it has been conducted with Professor Malin Bellman from Carnegie Mellon University, and more recent work is conducted with my PhD student at Ben-Gurion University, Michal Tenzer. And the title of the talk is an integrative approach for understanding the psychological neural basis of congenital prosopagnosia. So before even starting to talk about prosopagnosia and experiments and data, it is important to think why is it at all interesting to study face perception. So face perception is probably the most developed visual perceptual skill that we have as humans. Faces are actually all very similar in terms of the visual appearance. We all have two eyes above a nose, above the mouth. So faces are very similar physically, but despite its very basic visual similarity, faces can still convey a large amount of critical information such as age, gender, emotion, information about eye gaze, and of course information about identity. That will be one of the focuses for today's talk. Also, our face recognition abilities are largely immune to even quite drastic changes in viewing conditions such as illumination and viewpoints, and even more specific to faces, changes that are induced when with age and with facial expressions. And finally, faces are also very important for us for social reasons. A lot of our social communication is done through looking at faces and understanding the meaning of the expressions and the emotions portrayed by others. Faces are also very interesting from the neuronal perspective. It's been known for more than 30 years that there are face selective neurons in monkeys' occipitotemporal cortex, and this is an old picture taken from a paper by Bruce, Desimon, and Gross in 1981, and there have been many replications of this basic finding of cells that fire more strongly or in high frequency when monkeys see images of faces, of monkey faces or human faces, and reduce the firing rate for other stimuli. And more recently, there are very interesting studies using functional MRI in monkeys. Doris Tsao is one of the pioneers in this line of research. And in this approach, she's actually using fMRI to look for face-selective foci in the monkey brain, and then record electrophysiological responses from cells within those specific patches. So this is another very interesting approach to study faces. In the human brain, it's also been known for many years that specific lesions to occipitotemporal cortex leads to specific level of face perception abilities, and I will dwell on that in a few slides. This impairment is called prosopagnosia. And there are many studies, many of them were done by Shlomo Bentin, showing selective ERP components for faces compared to other stimuli, known as the N170. And again, a lot of functional imaging work that showed space-specific activation in the lateral occipital cortex, in the superior temporal sulcus, and in the fusiform gyrus. And the activation in fusiform gyrus is very consistent, probably the most consistent focus of these three, and has been replicated many, many times since the initial finding reported in 1997 by many labs using various stimuli, so it's a very robust effect. In recent years, people have started to shift their thinking about face perception and suggested that face perception, or that we need to think about face perception more of the depends on the distributed activity of many regions in the brain, and not only the occipital-temporal cortex or the fusiform gyrus, as have been suggested before. And these are images or pictures taken from two papers, one by Alumiti Shai and the other one by Corbini and Huxby, showing or trying to depict this notion of a face network. And I will focus on this diagram because I'm going to use it in the rest of the talk to illustrate several points. So their suggestion is basically that we should think about faces as being processed by two separate systems. One is a core system that is located in the visual cortex or occipital-temporal cortex. These will be these regions, and it's face-specific, and it relates to the visual appearance of the face. The other system, the extended system, is activated by faces, but it's not essentially face-specific in the sense that it will be activated by other stimuli, and it is involved in things like retrieving the emotion within the face. So regions like the amygdala will be part of that network, and it is also related to information about the person, or person knowledge as they term it. And these regions will include the anterior paracingulate and posterior cingulate cortex, regions within the superior temporal sulcus, the anterior temporal pole, and these are the regions that will be activated by biographical knowledge, by episodic memories related to the face. So again, it's important to stress that these regions will also be activated, by example, by familiar voices, but still they are part of the face processing network. So our approach for studying this network was to actually focus on individuals who are impaired at face recognition. From the sort of classical perspective of neuropsychology, study the impaired brain in order to infer about the normal processing. And the first group of individuals in that context are, of course, those with acquired prosopagnosia. So these people have an impairment in face recognition, obviously, and this impairment is a result of a specific brain injury to regions in occipital-temporal cortex, and so it has a late onset. They were perfectly normal before the onset of the lesion, and following the lesion, they became impaired. So this approach, again, as in classical neuropsychology, is very useful in order to understand regions that are necessary for face processing, because we have a lesion, the specific region in the brain is no longer functioning, so we try to deduce from that what is the role of this region. But this approach also has some limitations, specifically because we cannot control the location of the lesion and the extent of the lesion, and it's hard to compare across subjects because of the variability. And so more recently, a second type of impairment in face recognition became sort of more popular, which is termed congenital prosopagnosia. And again, one of the first studies was conducted by Shlomo Bentin and Leon Deuel. in 1999, I think it was published. And these individuals have a fairly similar behavioral profile to individuals with acquired prosopagnosia, and I will talk a lot about their behavioral profile. However, they have no known cortical lesions. So if a radiologist would look at their MRI images, they will not be able to tell that there's something wrong with this particular individual. Their brain, from every possible perspective or regular types of imaging that were conducted, looks completely normal. And so as far as we can tell, their impairment is actually present from birth, and it also tends to run in families, and there are several groups now that look at the genetic pattern of heredity. And at the beginning it seemed to be quite rare, but as we go along, and other groups as well, it seems that now people talk about prevalence of about 2% of the population. So it's quite a lot. It's not as rare as we initially assumed. And studying these individuals is interesting from the theoretical perspective that I will try to talk about, but in terms of the neural underpinnings, it's very useful in order to understand which brain regions actually participate in this big, large network of face perception. So I'd like to read two quotes to you, taking from descriptions of two of our participants, just to give you a flavor of what it's like to have congenital prosopagnosia. So this is a relatively older female subject from the US, and she spent several days in our lab, and then she returned to her home, and she sent us an email saying, "I'm glad I was able to spend some time with you. It's nice that now when I write to you, I can picture you in my mind. Well, picture you may not be quite the right word. I just realized my mental picture is medium height, slender build, longish black wavy hair. Face, well, it's there. 2 eyes, a nose, and a mouth." So this woman is clearly not blind. It's important to stress that she can easily see that the face. She can recognize the features of the face, but somehow these features are not glued together to form a unique identity. And as can also be seen, she relies on other cues such as body build in order to try to recognize the people that she sees, but obviously these are not enough. Another description is from an Israeli participant, a 60-year-old female, and this is taken from an interview we had with her. She remembers that at a young age she was not able to recognize her parents among other people. This created much anxiety and fear to be lost in a big crowd without being able to recognize her parents again. She describes that she usually recognized her parents by external features such as glasses or hairstyles. She remembers a very traumatic event related to that. At the age of 5, her mother drastically changed her hairstyle. Although she was with her at the time, she misrecognized her for another woman, and she remembers the panic she felt when realizing she approached someone else, and the fear when understanding that her mother had changed. This fear continues to exist throughout her life, and therefore she feels that she develops good auditory memory and attention to clothes, voice, and other details that may be useful for recognition. So again, the basic picture is that she can recognize, she can see the face, she knows it's a face, but she can misrecognize even a very, very close person such as her mother, and she relies on external cues such as glasses and hairstyle for recognition. I will talk about the behavior in just a second, and there are many experiments to explore. So that's it.
Did you ever ask her when she became aware of that and the fact that other people recognize faces?
So they usually understand at a very early age that something that they don't recognize faces so well. They have early memories of such experiences.
No, but other people do.
I think that for some of them actually say that they thought that everybody's like that, and some of them realized at some point that something is different about them. But for some of them, because many of them didn't know such a phenomenon existed. So they thought, well, everybody just recognizes faces the way they do. So not all of them actually realize that there is a deficit. They know something is weird, but they don't always understand that it's a real deficit in them compared to others. Okay, so now to the formal experiments. These are their own descriptions, which are very interesting and very important for us. So the outline for the talk is I will start with describing their behavioral profile and then move to the neural part and trying to see whether we can understand the neural underpinning of this impairment. So in terms of the being included in our participant pool, what does it take to be a prosopagnotic? So our inclusion criteria is the following. We have several different tests that include face recognition, face perception, and memory. And participants have to be significantly impaired on at least 2 of these tests. We calculate the significance by using these scores compared to large control groups. But as I will show later, we also use age-matched control groups when we want to compare specific effects within the prosopagnotic groups compared to controls. And there are various ways that people use for diagnosis in the literature, and we kind of try to be consistent with what other people are using. So our first test is fairly simple. We show them images of faces and they have to tell us who that person is or tell us something about the context. They have to be quite specific in terms of the context. It's not enough to say an actress. They have to give us some more information. Or they can say they don't know who it is, or sometimes they give an incorrect responses. And this is a table taken from a recent study. We have 14 subjects here. You can see their age, gender, and you can see the raw data, the accuracy on this score, and their normalized Z-scores. And it's quite obvious that 12 of these subjects are clearly very impaired, and the other 2 are borderline in terms of their responses. You can see the average performance of control is around 85%, and the average performance of the prosopagnotic group is around 45%. Now, we run several different tests, so even though this person here is not drastically impaired on this test, I can assure you that he is drastically impaired on at least 2 other tests. So this is supportive, of course, but it's not enough, and I'm not going to show all the diagnostic tests, so you'll have to trust me on that. The next thing is to see whether there's any impairment in memory and perception of unfamiliar faces, so to make sure that it's not just retrieval of long-term memory and long-term information. This is a test developed by Brett Duchesne that became fairly standard, and so we also use that in our lab. Basically, subjects are asked to memorize faces, and then they are shown a series of faces, and they have to detect which one was in the sample that they had to memorize. And it has several stages of increasing difficulty, and the results are here. So again, most of them are showing drastic impairment in this test, which is, just to remind you, this is short-term memory for unfamiliar faces, so it's a perceptual memory, perceptual difficulty, not long-term memory. And our next test is focused only on perception with no component of memory, and here subjects are shown an upright face and 2 target faces, and they have to decide which one is the match. And again, you can see the interesting results here are in terms of reaction time, because the faces are shown for an unlimited exposure duration, and you can see the average performance of the CP groups is more than almost 3 times higher than that of the control group. But they are very, very, very slow, even if they eventually manage to produce the correct response. So we have impairment in face recognition, in face perception, in face memory. And what we wanted to ask then is whether there's a more general impairment that maybe could account for the difficulties that we see in various tasks related to face processing. And the usual suspect when we talk about face perception is, of course, holistic processing. And what I mean here by holistic processing is your ability, hopefully, to immediately see a face in this image, even though it is actually composed of many different vegetables. Most people would quickly see that this is a face, and just to test you, this holistic immediate perception is lost when the face or this image is inverted. So what we mean here by holistic perception is the ability to immediately glue the features comprising the face and see the face. And so the first test that we used was to use the unfamiliar face perception, but now use the inverted version to see whether we get the classic inversion effect, in which most subjects show lower performance for inverted compared to upright faces. And so this is the result in a large control group. You can see a nice inversion effect in terms of reaction time, so longer reaction time for inverted compared to upright. And these are the results for matched control groups to the prosopagnosic subjects. And the result in the prosopagnosic group is actually very different. We can see that they're quite slow on both upright and inverted faces, but they show comparable performance for both upright and the inverted faces. So no evidence for the inversion effect that we clearly see in control. So this is one supporting evidence suggesting that indeed there's something wrong with their holistic perception of faces. But we wanted to take it even one step further and use a classic task that was designed by Young in 1987. This is the composite face discrimination task. And people have used it since many, many times, and it's sort of considered the gold standard of testing holistic face perception and holistic perception in general. And in this task, the stimuli are built like that. So there are two halves of the faces, the top part and the bottom part. And in this condition, the top and the bottom part are aligned. So they look as a normal face. And the top part of the face could be either the same or it could be different. And the bottom part of the face is always different. And the subjects are instructed very explicitly to perform the same different discrimination task based on the top part of the face, and we tell them that they can basically ignore the bottom part of the face. Now, because face perception is holistic, it is very difficult for most people to ignore the bottom part of the face and to perform the task only based on the top part of the face. So people tend to make mistakes, even though this seems like quite a fairly easy task. Now, the second condition is the misaligned condition, and we use this condition in order to impair the holistic percept of the face. So in this condition, people actually make fewer mistakes compared to the aligned condition. And this is the full design, and the conditions of interest are here. When the top part is the same, we compare the aligned to the misaligned. And the prediction is that because of the holistic impairment in face perception, our individuals with congenital prosopagnosia should actually outperform the controls. So again, these are the results in a large group of controls in terms of accuracy. So we see a nice composite effect, better performance for the misaligned compared to the aligned faces, and this is in the matched groups. But again, in the CP groups, we see no evidence or much reduced, non-significant effect of the composite manipulation. So the performance here is not different in the misaligned compared to the aligned condition. And the same result is also evident in terms of reaction time. So controls are showing longer reaction time for the same aligned faces, and the prosopagnotic subjects again do not show any difference here. So this is quite a dramatic, if I may say so, demonstration of their impaired holistic perception, and it is dramatic because although they are the so-called patients, they actually perform better than the control. So you can see here that their performance is actually significantly larger here compared to the controls. And so our next move was to see whether we can tap this impairment in holistic processing in tasks that do not involve faces. Is it a more general deficit in holistic processing that will be evident in other tasks as well? And here we also use the classic Navon stimuli. Subjects are shown letters, large letters that are composed of small letters, and the small and large letters could be consistent, like these two, so small S's that make an S or an H, or inconsistent, so small H's that make an S, and the inverse here. And in some blocks they are asked to judge the global shape, and in others they are asked to judge the local shape. This is the result in the large control group, and let me also show you the matched control group. The basic finding, and the one that has been replicated many times, is that for most people it is actually much easier to perform the global task. So we see sort of the forest before the trees. As we can see here, the global performance is faster than the local performance, and the interference here from the global element to the local element is greater. And the result in the prosopagnotic subject is strikingly different. It's really the inverse pattern. So the prosopagnotic subjects are showing much slower performance for the global compared to the local, and as you can see, this is really the inverse, and showing a huge interference in the other direction. So there is a local local elements interfere much more when they have to judge the global shape. So we can see evidence for impaired holistic perception with faces and with non-faces, and of course the question is whether there's any correlation between performance in these tasks. So this is a correlation between the performance and index calculated for the performance on the Navon letters and the composite task, and you can see a modest correlation. It is a trend of 0.06, but with a group of 14 subjects, I think this is quite nice, showing that the more local you are on the global local task, the Navon task, the more local you are also on the composite task. Now, what is also nice about the composite task that it actually correlates with the actual performance of our CP individuals on other face perception tasks. So this is, for example, a correlation between the performance on the famous faces task that we give them, so how bad they are on recognizing famous individuals, and it is negatively correlated with their index on the composite task. So those that are worse on face recognition are also more local on the composite task. So it has some the composite task has some relation to the way they actually perceive and process faces, and this is also true for the short-term memory face discrimination task. Again, a negative correlation with the reaction time.
Is it also true for the controls? That is, I mean, there's some variation in the controls in the local global task.
There is some the problem with the controls is that they are very good on the face processing task. So you almost always, you have the ceiling, and then you cannot show a nice correlation. But there's also some variability in control. It's true. It's not that everybody is great. So the summary of this part of the behavioral finding is that we can show impaired recognition of famous faces in the prosopagnosic subject. We also see impaired perception and memory of unfamiliar faces, so a perceptual problem. And we see impaired holistic processing in both faces and non-face stimuli, and the correlation I just show you between them, and also the correlation in performance of face tasks and the composite tasks. So we think that they are actually meaningful. So the next question is, of course, what gives rise to this dramatic impairment in face recognition in terms of the neural architecture of the face processing network in the brain of the individuals. So let me go back to this diagram, and just to remind you, these two systems, the core and the extended system. And our initial attempts, as well as the attempts of others, were focused on investigating the core system, because this is the most intuitive and straightforward hypothesis would be that if these regions are necessary for face recognition, as we know from acquired prosopagnosia, there must be something wrong or different in these regions that will be evident in individuals that are so impaired in face recognition. So at least our attempts to show any differences in the core systems were actually negative, so we continue to show in different studies and using different paradigms that there are no structural alterations in the core system, and also that there are no functional alterations. I have to say that some people, some of them are actually in this room, showed some different results, and actually did find some differences also in the core system, but at least our findings...
It's not mutually exclusive at all. It only proves that the FFA, for example, is not a sufficient condition. It still could be a necessary condition. So finding that the FFA is not selective in one patient and it is selective in another patient means that it is not sufficient.
Absolutely.
But also may mean that it is necessary.
No, I totally agree with this interpretation. This is exactly what we also suggest. That the conclusion from the fact that we don't see these alterations, or if there are occasional alterations in other participants that we've not tested, suggests the necessity of this region. Yeah.
So other cases, like two patients where perception is normal but only memory.
Yes, there are such cases of individuals that have what people term prosopagnosia, so it's more a memory-related problem in face-specific memory problem, but it's different than what we're looking at here.
In this case, in your case, it's not clear. So it's just memory perception problem. You probably cannot memorize.
Yeah, but I mean, they don't have a general memory problem. They can memorize other things. So it's yes, but it's because we see a perceptual problem. We think it's a prerequisite, right? So we have to have good perception for faces in order to be able to eventually memorize them. But the problem starts earlier, so it's not a memory problem, but a perceptual problem. That's at least our interpretation. Okay, so this is where we were or still are, and our sort of current attempts are more focused at looking at possible alterations in the extended system and the connections between the core and the extended system, because sort of our working hypothesis that if things are okay here, but then face recognition is impaired, there's probably something wrong with the transformation of the information from this core system to the extended system. So our sort of most recent experiment that is conducted in part of a larger project, but also yielded some interesting results, is actually a very, very simple experiment in the larger context, we just use it as the face localizer. So we're showing subjects blocks of emotional faces, neutral faces, famous faces, unfamiliar faces, and buildings that we use just to contrast to be able to localize the faces. And the goals of this particular design was first to try to replicate the normal activation in the core regions using various face types, so emotional and famous, and as I mentioned, to examine the extended face network and the connectivity between the core and the extended networks. Okay, so first of all, can we replicate? So these are the results from a group of 7 age-match control subjects, and these are the regions in the core system, the occipital face area, the part of LO that is face selective, posterior superior temporal sulcus, and the FFA, the fusiform face area, all shown very nicely here, and also in the prosopagnosic subjects. You can see all these foci here, and also you can see them on an individual subject level. So this is basically a replication of our previous studies with a set of additional 7 subjects, which is also important or reassuring. And let me just mention that according to the Gobinian Huxley model, these core regions are thought or to be related to what they term extraction of invariant features or properties of the face for identification. So at least in terms of the fMRI signal, this region seems to...
just to say all, I think it is worth mentioning a very nice result, which is evident here and fits literature very well, which is that the CPs are more bilateral than the controls. So in the controls you have much more involvement of the right hemisphere as opposed to the left, whereas in the CPs you have a bilateral activation. And this fits very well both clinical findings and many other findings in literature, which might suggest that the way faces are processed, as you said before, is different. And the fact that they are less configured, less global, activate the left hemisphere more than in normal controls that activate the left hemisphere less. And this is very nicely seen here, I think, and fits literature very well.
I'm not sure it holds if you count the number of voxels in each individual and compare it, but I agree that it does look like that in the image, but I'm not sure the statistics hold in that respect. But it's true that there is certainly a trend here. This is all the faces that we've used in the localizer compared to the houses. So just the big chunks of green activation is for the houses. Okay, in the parahippocampal gyrus and PPA and probably V4. Okay, so just to show you the activation that we extracted from the FFA, the right and the left. This is in the matched control group, and this is in the CP group, and as is evident, the activation is really very beautiful, and there are no statistical differences in terms of the activation between these two control groups. And in the right FFA, we see both groups actually an effect of familiarity. So the famous faces elicit a stronger signal compared to the unfamiliar faces. And we've done the same analysis for the other foci as well. I have them at the end of the talk. I can show them to you, but for the sake of time, we should move on. So what about the extended regions? We can show normal activation in the core regions. What about the extended regions in the posterior cingulate, in the anterior medial prefrontal cortex, in the anterior temporal cortex, and in the amygdala? These are the regions that I'd like to focus on now.
The effect that you mentioned in the amygdala, is it left to the right or to the right hemisphere?
I will show the data in a couple of slides. Okay, so for the sake of the posterior cingulate and the anterior paracingulate, I have to quickly go back to an older study in which we also use famous faces. And in this study, when we contrasted famous faces compared to unknown faces, we found nice activation these are two different control groups, in the anterior paracingulate cortex and in the posterior cingulate cortex for familiar compared to unfamiliar. And in the prosopagnosic subjects, when we used the same statistical threshold, we were not able to show any activation in these regions. And even when we lowered the statistical threshold, we got some activity in the posterior cingulate, but none in the medial prefrontal. And these are the regions that are marked here in the model. So the anterior paracingulate and the posterior cingulate. The posterior cingulate is thought to be related to maybe retrieval of episodic memories, and the anterior paracingulate are thought to be related to extracting personal traits and other features of the related to person knowledge. So
as well as selective attention.
Sorry?
As well as selective attention, which might be a factor. The anterior cingulate is the main region in the brain that is involved in selective attention. And it could be in a way that it is important to say that these regions are activated by many stimuli and in various paradigms. So, as I said before, it's not selective to famous faces by any means. It's not what I'm saying. And of course, attention is also part of that. So, it could all be related, but just in the context of this particular contrast, we were not able to show activation here. And perhaps if we were to use other stimuli, non-face stimuli, probably these regions would be activated. It's not that they're not functioning in these individuals. So, what about the anterior temporal cortex? The anterior temporal cortex is a very interesting region from the neuropsychological perspective. We know that lesions in this region are also involved in acquired prosopagnosia and are related to impairments, specifically in impairments in recognition and retrieval of person knowledge. And this region is also very difficult to image for technical reasons. So there are many artifacts in this region, and it's been difficult to get good activation for several years. And using this particular localizer, we were actually quite lucky. And this is an image from our control group. You can see very nice activation in the anterior temporal cortex, here in the right anterior temporal cortex. And again, subjects are also showing this activation on the individual subject level. But again, when we look at our prosopagnosic subjects, we see very, very little activation. This is the same region here. And this activation is contributed by one or two of the subjects. It also shows some noisy signal, as I will show you in a second. So we have difficulties in activating the precuneus, posterior cingulate, and anterior cingulate, and also the anterior temporal cortex that is part of the network involved in extended face processing and specifically thought to be related to biographical knowledge related to the face. And looking at the time course of this region from the matched controls, this is what we see here. So the signal is reduced compared to what we find in FFA. In the FFA, I probably don't remember, but it was more than 1% signal change. Here it's around 0.5, but still nice and significant. And in the prosopagnosic subjects, because they don't see this activation, what we did was to sort of force the activation foci from the controls on the prosopagnosic subjects and sample the profile just to see whether there's anything in there. So when we do this, we get this quite noisy and weak activation. And of course, this is significantly different from what we find in the control groups. So this is consistent with the fact that we don't see significant activation in this region. As mentioned before, the temporal region, particularly the temporal probe, is more related to semantic and particularly verbal semantic actually. So perhaps a better test would have been to activate or to see activation in controls and CPs in tasks that do not require identification of the base of face matching or any type of other activity that would, in my opinion, would show no activity in controls as well as in CPs. So I think that what we see here in controls is actually not related to face perception, it's related to like frontotemporal dementia. That's one of the aspects of frontotemporal dementia. What you see here, the activation is related to the extraction of information from semantic memory, rather than processing the face itself.
So during the task, maybe I didn't mention it, during the task what they had to do is the one back task, just to keep them awake in the scanner. We didn't ask them to recognize the faces, and so, I mean, recognition was not explicitly required. People do this automatically, but at least they do the one back task, and they're not focused on recognition.
Can they do the one back task?
The prosopagnosic subject, they do it, but they're actually worse than controls. But even when we use tasks that are equated, here we didn't equate performance. But, yeah, maybe we should do a task that is only semantic and not visual.
The identification of the individual face is different in normal.
Yeah, I agree. But at least it means that when you cannot identify, this region is not activated. So again, I'm not saying that this region, there's no, I mean, the region is structurally intact, there are active neurons there, I'm sure. It's just that using this particular task, we cannot show the signal in this region. So what about amygdala? So this is actually an interesting control region because both other groups and also our group actually showed normal emotion recognition in prosopagnosic subjects. So the question is whether all regions in the extended network are created equal such that they will not show normal activation, or whether particular regions that are important for example, emotion recognition, such as the amygdala, would actually be normal. And this is a strong sort of test for us. So this is the activation in the right amygdala in the control group, and we can also see very nice activation in the CP group. Again, in terms of anatomy, it's the same place, and as I mentioned, this is consistent with their normal behavior on this task. So it's not that our protocol is wrong for some reason, it's not that their entire brain is different, it's quite specific to the network that is involved in identification and person knowledge. Same contrast. No, we use the same contrast, so this is the time course. So we see somewhat stronger activation for the emotion compared to the neutral. It's not significant, but we see the same trend in the two groups. But it's not a huge effect, I have to say. I think our faces were not fearful enough, really. We chose faces that are angry and fearful to be able to activate the amygdala, but I think that... Sorry? We didn't test them on... One of them was in our original test, and he was fine on emotion recognition, but we didn't test the other, not explicitly. So I don't know. But anyway, the amygdala seems to be normal in this group. So the next thing that we examined is actually the connectivity between the regions. So we keep saying that maybe something is wrong in the information propagation from the core regions to the extended regions. So it's time to look at that. And the way we did it here, and I have to say this is still somewhat preliminary, so we extract the activation profile from each of the ROIs, from each region of interest, just the activation profile along the entire experiment, and then we do it separately, of course, for each individual, each region of interest, And then we calculate the correlation, coefficients across all regions for each subject, and then we average each of the groups. And after we do that, what we get is the following. So we get a matrix with all the regions that we pre-selected. It's important to say these are only the pre-selected regions. And the color indicates the level of correlation, so the red is maximal correlation. And these are the regions...
It's correlations in the average time course between one region and another.
Yes, yes. But it's the average along the entire experiment, so it's not just a peak activation from particular conditions. We just take all the time course and correlate all the time course. So what we're looking at here is the matrix that we got from this group of 7 age-matched controls. And these are quite small, but this is the SFA, the fusiform face area, right and left, the occipital face area, the LO, and superior temporal sulcus. So these are actually the core regions, and this is in controls, and this is the matrix that we get in our prosopagnotic subjects. 7 CPs. And both visually and our statistics suggest that there are no obvious differences in here, but as I mentioned, this is still work in progress, and we're trying various methods to look at the differences here.
What is the threshold value that you could between two ROIs such that you define it as a true connectivity, as a true connection?
We don't threshold here. We use whatever value we got. I'm showing you so for each subject we calculate the correlation between each region, all pairwise correlations, and then we average across all the subjects, and then we have all these values. This is not threshold. The threshold is we do not implement any threshold here. So we just want to see all the correlations that exist. Okay, so the level of correlation is, of course, indicative of how significant it is, but this is not threshold. This is the real raw data.
I'm confused, though. Perhaps I don't remember closely, but it seems to me that Correct in a recent paper showed problems of connectivity between... I'm wrong?
No, so I will show the data in a second. Okay. This is a structural connectivity. Yes. So this is just functional, just looking at how well two regions are activated together. So how similar the activity in one region is compared to another region. Strictly functional. Yes, only functional.
This assumes that the delay in each region is the same.
It's true. Yeah, there are many assumptions, but this experiment is a slow block design experiment. So there's nothing fancy about timing here. It's really, blocks of 10 seconds. And so we think timing here is, timing is important, but in this particular experiment, I don't think that we can actually relate to time shifts between different regions. But it's true that there could be a different hemodynamic response in different regions, absolutely. Yeah.
Looking at statistics, patients, instead of averaging over the results of all of them, does it change the picture in some way?
If we look at statistics?
Yeah, when you average the whole group, and maybe each one is impaired sort of differently.
So when you look at the individual images, you see sort of the same pattern. There is some noise, and what we actually try to do now is to use clustering algorithms to see whether we can separate them or not separate them using other approaches rather than just looking at the correlations and looking at statistical differences of these correlations.
I understand here that using the term functional connectivity is quite misleading, because what it is the similarity of the response. Nothing, not necessarily connected to connectivity at all.
So there are different ways to look at connectivity in terms of function. I should say probably effective connectivity. This is probably more accurate. Okay, so at least in the imaging literature, people, of course it's not connectivity. We don't know whether there's a fiber going from one region to the other and transferring the information from this analysis. We just know that they function in a or respond in this particular experiment in a similar fashion. That's
functional correlation.
Or functional correlation or effective connectivity. I'm not saying anything about timing here, really. Okay, so this is the core region, and then just to focus your attention, this is the amygdala, so part of the extended network, here and here. And surprisingly, the amygdala seems to be kind of not very highly correlated with the activity in the rest of the core system, but if anything, it is slightly more correlated in the CP group than in the control. And the next region that I'd like to draw your attention to is the anterior temporal cortex, in which you can actually see these nice correlations and the complete lack of correlations in the CP group. So as I mentioned, this is just preliminary, but it just confirms our sort of working hypothesis and results from different approaches that I will describe in a second. And this is just another region, another sort of network that we've looked at. These are phase building selective regions in the parahippocampal place area and in the transverse occipital sulcus. So we just looked at another network to see whether there's any general effect or any sort of more general differences. And again, this seems to be similar in the two groups. So just to summarize this, we see normal activity in the core phase network, and we see normal connectivity in the core phase network, and some evidence for or some hints for disrupted propagation of information to the extended network that is involved in person recognition. So, question?
Yes, I actually have a question. The question is about anatomy. So you use functional localizer throughout, right? So how much of how blurred is your anatomical resolution? So you say, I don't know, temporal lobe, anterior temporal lobe. What does it mean?
So our functional imaging is done in 3 by 3 by 3 millimeter cubes. This is
but you use functional localizers to begin with, right? So that
It's different.
So when one subject to the other, right? So like when you use a group, you don't really know, whatever the size of your voxel, you don't really know that it's exactly the same anatomical voxel.
no, I can only. It's true,
The
but
question is, I'm not saying it critically. I'm saying it's a question. How much of an anatomical location do you actually identify? So when you say anterior temporal lobe, what do you mean?
so we normalize all the brains to talaric space,
Sure.
which is sort of the best you can do. And in terms of the coordinates, it's within, of course, some small standard deviation. It's in the same location. But I have to say that in a different study, we actually.
Standard deviation?
I can't remember, but it's reasonable. I mean, it's reasonable. It's in the order of what people report in fMRI literature. I can't remember the numbers offhand, but it's reasonable. So it seems to be well located in everyone. Okay, so
That's true across all the ROIs?
So just to finish the story, we've talked about functional connectivity, and of course there are some critical and there were some questions here about this approach, which are completely justified. So the other approach is to look at structural connectivity, which is not related to function, so has some limitations in that respect, but at least you know that you look on fibers. So this is one thing that we do that I will describe briefly, and I will try to end with showing some possible behavioral consequence of this disrupted network.
In relation to the green summary, so with the various experiments, you can try to look for the direct contrast, so you can show us like the mass of the normal and the mass of the congenital. What about contrasting
So because it's a group of 7 CPs and 7 in other experiments that I tried, it didn't work so well. In this one, I actually didn't contrast with another recipe, but I can do that. In previous experiments, when I tried to contrast groups directly, it didn't work so well. Very nice.
I don't see any difference between looking at the group of 7 and doing the contrast of 7.
So you want to see if there are differences?
Right.
I can do that. Easy. Okay. So looking at structural connectivity. So of course there are inherent problems in the functional connectivity, and also particularly when you're not able to image particular regions. As I showed you, it's hard to talk about functional connectivity of regions that are not activated, right? So we have to look at structural connectivity. And a few years ago, we ran a DTI study, diffusion tensor imaging experiment. And in this study, we actually looked at specific fibers that connect the fusiform gyrus, where the fusiform face area is, to the anterior temporal cortex. So this is the inferior longitudinal fasciculus, so ILF. And the second fiber that we were focused on was the inferior fronto-occipital fasciculus that connects the fusiform gyrus and other regions to the frontal cortex. And our approach here, again, was very sort of focused on these particular fibers. Whoever tried to use DTI knows how many variables there are and how you can do the analysis in many different ways. And we had a very focused approach here. And so using these fibers, what we showed is that fractional anisotropy, the measure of the sort of structural integrity of the fibers, was actually significantly reduced in the CP individuals here in gray compared to a control group in both fibers and in both hemispheres. And importantly, this was also correlated with the behavior. So poor structural connectivity was also related to poor recognition performance. And another aspect of this is that, again, the damage or the difference was specific to these particular fibers. So when we use other fibers as controls, like the forceps minor and the forceps major, we actually didn't find any difference. And this is particular forceps major is part of the visual system, so it's also an important control to show that it's related to these fibers and not some general phenomena. Just another part of work in progress, following our functional imaging results, we actually also looked at another fiber tract, the cingulum, that connects the anterior and posterior cingulate, and also showed some preliminary evidence for a difference in the CP group in the left hemisphere compared to the controls. But again, this is sort of work in progress, but again makes sense and sort of nicely tied the functional and the structural results. So let me just end by looking at part of a behavioral result that we had that is, again, consistent with the idea of disrupted network. And what we looked is we looked at implicit familiarity processing with the logic that if there is some representation of the faces is fragmented because of this disconnection, maybe these individuals would have some sort of rudimental information about the identity of individuals that can only be exposed implicitly but not explicitly. And in order to look at that, again, this is the experiment that I showed you earlier in relation to the anterior and posterior cingulate. We used a set of faces and unknown faces, and the subjects, these were brief trials, and subjects were instructed to say whether the second image is of the same person as the first one. So this would be, the answer here would be yes, and here it would be no, and here it would also be yes. And this is sort of the interesting and more challenging condition. And in order to to look at the question of implicit processing, what we've done was to take the responses of our subjects and parse them based on the response in the questionnaire that we give them. So as I showed you at the beginning of the talk, we explicitly show them images of faces and ask them to say whether they recognize them or not. And so we can take a set of images of famous faces that we show them in the experiment and parse them into famous faces that they actually explicitly recognized when asked, or faces that are famous and should be familiar to them, but they do not recognize them explicitly, or faces that are truly unknown, so they're not supposed to recognize them. And because of repeating effects in the control group, the same analysis was only possible in a 5 out of the 12 controls that we had in this experiment, where performance was not as ceiling for face recognition. And so when we look at these 5 controls, and this is the interesting condition where we were repeating the identity using 2 different images, we see this nice effect of maximal accuracy for faces that are explicitly recognized, and then the lowest accuracy is for faces that are truly unknown. But the response for the faces that are famous but not explicitly recognized is sort of in between, both in accuracy and in reaction time. And interestingly, a similar pattern was also observed in the prosopagnosic subjects. So here you can see the drop is much larger for them, but the trend still exists in both groups. So just to summarize everything that I told you, we see evidence for impaired holistic processing across tasks in individuals with congenital prosopagnosia. We see normal fMRI response in core regions, and we see evidence for disrupted connectivity in terms of the fMRI signal and the white matter connectivity that goes along the same lines. And we see some behavioral evidence in the form of implicit face recognition that is consistent with the interpretation of having a disrupted network. So our general conclusion is that congenital prosopagnosia can be thought of as a disorder of disconnection rather than as a result of a localized lesion, and this is of course different from the approach regarding acquired prosopagnosia. So I think in terms of theory, it is interesting whether these 2 impairments are the same or different, and whether a similar behavioral profile could actually result from a different neuronal underpinning. And I'd just like to end by thanking other people that were involved in various parts of this project, and just to mention that this is a collaboration with Professor Leslie Ungelal, who is doing similar experiments with primates fMRI to see homologies between primates and us. And thank you for your attention.
Time for some questions, if anyone.
No more questions.
Maybe a comment regarding the connectivity. There is an area which is recently much expanded, and many more papers are using this, which is called functional connectivity during rest. This, unlike the expansion correlation or activation, there is some mathematical models which show really why you see some synchronization activity, or not, it's not activity, synchronization fluctuation of the signal, and this can be used with not only the ROI which responding, so this could be a way to...
So we actually, in the same scanning session, we also use resting state, and we have this data, and we are still busy analyzing them, but certainly show some interesting patterns. But I have to say they go along the same lines with what we see in the activated state.
Not exactly. I go with the same line.
Yes. No, it's a different approach, and it's an interesting approach, and But at least from what we've analyzed so far, it seems to be consistent with the rest of the story. So this is also sort of supported.
Just to make one additional comment very short. In acquired prosopagnosia, also there are studies showing the temporal connectivity. So the dissociation here is kind of fake, because in acquired prosopagnosia, the set of vision usually is big. Usually you are talking about CDAs, and therefore it is very difficult to disentangle very thin localization as opposed to connectivity. However, Bonarsion's famous case DS shows very clearly it's a problem of connectivity, and so there's a new study by Barton that shows the same things. Connectivity probably it's a major factor in prosopagnosia, which makes sense because in prosopagnosia by definition, particularly associative type of prosopagnosia, not a perceptive type, is basically a problem in connecting a particular face with a particular type of semantic information related to it. So it makes sense that it's a problem of connectivity.
I should probably phrase it in sort of at least more. The recent approaches indeed that even acquired prosopagnosia is more of a disconnection syndrome, but at least the classic approach related to it more as a localized lesion rather than being a disconnection. So that's the contrast that I prefer to put at.
I want to raise another issue. I mean, you sort of ruled out the possibility that the problem may lie in the core areas because you didn't find any difference between your prosopagnostics and the normal control. But the resolution you're looking at is you were looking at the average activation in a region of interest. Now, it may be worthwhile, you may get the same result, it may be worthwhile to start looking at using, distributed representations of, voxel representations and seeing if you have any information there. And so that's for the core areas, but even you could also look at across all areas and see, okay, do you see any difference between the prosopagnostics?
So it's part of what we're trying to do with this data set that is actually quite useful. I mean, it's enormous, so you can do various things, and it's one of the things that we want to do, or we're sort of on the way.
So has anyone tried this? I mean, it looks something like Griggs called the
with prosopagnotic subjects?
Yeah.
No. People use this approach in normal subjects, but in an older study we did run an event-related fMRI adaptation study to look at specific effects, and even with this paradigm we didn't find any differences in the core system. But of course we have to look at the pattern.
But the fact that we could find selectivity in the FFA coupled with very, very profound prosopagnosia wouldn't imply that the FFA is not sufficient?
Maybe the average activation is let's think about this. Prosopagnostics have a similar average activation, and not only that, but if you look at their distribution of responses across the FFA voxels, it would be the same for different faces, while if you would look at controls, you find the distribution of the patterns are different for different faces or for different categories. And so you have some information there that you do not see in the average activation. That's what I'm.
And there are new approaches that people use, like using wavelets to look at different synchronicity and different frequency bands and things like that are sort of interesting. To see whether you can actually find some differences in that respect, and sort of on the way of trying to use that for our data. No results yet.
Any other urgent questions?
Urgent.
Okay.
Thank you
very much.